Good day, future doctors. So we will be continuing our discussion on the second part of the muscle tissue. This is the cut section of the skeletal muscle. As you can see, the muscle fibers are almost of the same sizes, and then they are multinucleated, and their nuclei are located at the periphery or underneath their sarcolemma. So let's say we have a patient who met an accident, and the patient sustained a tibial fracture. Part of the management is to put cast on the affected leg to immobilize it. So for the next few months, the muscles on the affected leg will not be used up, and they will undergo what we call the process of atrophy. So there would be a decrease in the size of the skeletal muscles. That's why after the removal of the cast, you will see the significant difference between the muscle mass of the left leg and the affected leg. And if we're going to view the skeletal muscles of the right leg under the microscope, this is what we will see. This is now the biopsy of the skeletal muscles of the affected leg of the patient. Can you appreciate significant differences between the sizes or diameters of the skeletal muscle fibers? Some skeletal muscle fibers have decreased in size, and you call that as the atrophy. The point is, you will never know that there's atrophy on this particular cross-section if you don't know how skeletal muscles would normally appear under the microscope. You will never identify what is abnormal if you don't know what is normal. There is that condition we call as acute myocardial infarction. We can simply refer to it as heart attack, and it can be because of the blockage or occlusion of one of the coronary arteries by the formation of atherosclerotic plaque. If we have excess cholesterol in our blood, some of these cholesterol molecules will be deposited on the wall of our arteries. The further deposition of cholesterol molecules plus the chronic inflammation will eventually cause the formation of atherosclerotic plaque. The plaque might cause total occlusion of the artery. One of the commonly affected arteries in atherosclerotic plaque formation is the coronary artery. This is the main blood supply of the heart, particularly its cardiac muscles. Should there be an occlusion in one of the coronary arteries, the cardiac muscles that are being supplied by the coronary arteries will undergo necrosis because they can no longer receive blood supply. This is the longitudinal section of the cardiac muscles. As you can see, the muscles are striated, they are branching and anastomosing, and they have those transverse lines which are representing the intercalated disc. This is how the cardiac muscles would look like in cases of acute myocardial infarction. As you can see, there is the infiltration of neutrophils indicating that there is an ongoing necrosis or inflammatory process. Again, you will never know what is abnormal if you don't know what is normal. Let's have a review. Muscles are composed of muscle fibers. If you group muscle fibers together, they will form muscle fiber bundle, or we call it as the fascicle. If we group fascicles together, then they will form one whole muscle. This is a whole muscle, and it is covered by epimysium. These are fascicles, which are composed of group of muscle fibers, and they are covered by perimysium. These are individual muscle fibers, and they are covered with endomysium. Take a look at one muscle fiber. It has a smaller subunit which is composed of filaments, and that smaller subunit is called the myofibril. This is one muscle fiber, and highlighted on the right side of the picture is its myofibril. I want you to take a look at the myofibril. You have their alternating dark and light bands. The blue arrow is pointing to one muscle fiber. This is the myofibril component of that muscle fiber. This myofibril has alternating dark and light bands. The dark bands are called A bands, and A bands are intersected in the center by the structure we call as the M line. The light band is called the I band, And it's very obvious in the picture that the eye band also has a line that intersects it in the center, and that line is called the Z line or the Z disc. What do you notice about the myofibrils? It has repeating structures. If an organ has a repeating structure, that repeating structure will now be referred to as the functional unit of that organ. So there is a functional unit in our skeletal muscles, and that functional unit is called the sarcomere. And take a look at the label in the picture. A sarcomere would range from one Z disc to the subsequent Z disc. So what does it mean? In one sarcomere, you have one whole A band in the center, and you have half of the I bands, because the sarcomere would range from one Z line to the subsequent Z line, and Z lines divide I bands at the center. The light band in striated muscles is called as the I band, and it is intersected in the center by the Z disc. The dark band is called the A band, and it is intersected in the center by the M line. The structural unit of striated muscles, such as the skeletal muscles and the cardiac muscles, are called the sarcomeres. One sarcomere would range from one Z disc to the successive or subsequent Z disc. So looking at that particular sarcomere, you will see that a sarcomere would contain one whole A band and half of the contiguous or successive I bands. So in striated muscle fibers, you have alternating dark and light bands. The dark bands are called A bands, while the light bands are called I bands. And I bands are intersected in the center by the Z line. We will discuss myosin, actin, and H band later. The sarcomere is the structural and functional unit of striated muscles, and it is usually the segment within the myofibrils that is between two successive Z lines. And as you have learned a while ago, you have there one whole A band and halves of the two contiguous I bands. This is just to prove to you that cardiac muscles also have their sarcomere. So they also have alternating I bands and A bands. Let's have another review. This is the I band, the light band within striated muscles, and it is intersected in the center by the Z disc. This is the dark band or the A band, and it is intersected in the center by the M line. A sarcomere would start from one Z disc to the next Z disc. So that's why you have there one whole A band and halves of the two successive or contiguous I bands. So I want you to memorize now the appearance of a sarcomere and the structures that you can find within it. This is one sarcomere. And take note that sarcomeres have two types of filaments. We have the thick filament, which is also known as the myosin. And it's very obvious in the picture that the myosin is situated in the A band. And the sarcomeres also have the thin filaments, and we refer to them as the actin. What do you notice? The actin filament is present in both I band and A bands, because there are portions of the thin filaments that are found along with the myosin in the A band. Enclosed within the rectangles are the I bands, and it's very obvious that the I bands only contain the actin filaments or thin filaments. So that's why this portion of the sarcomere would really appear to be lighter. And take note, the I bands are intersected in the center by the Z disc or Z line. And the protein present in the Z disc is called actinin. Again, the filament present in the I band is called actin. And the protein present in the Z disc is actinin. So do not be confused with the two terms. Enclosed in the red box is the A band or the dark band. And as you can see in the dark band, you have there the overlapping of the thick filament or myosin, and the thin filament or actin. And it's also important to take note that in terms of the thick filaments, there are thick filaments on both sides of the A band, and they are joined together in the center by the M line. These are the I bands, and the dark staining structures found at the center of the I bands are called the Z lines or Z discs. This is the A band, and then the structure that you can find in its center is called the M line. What I want you to appreciate at this point is that the central portion of the A band appears lighter than the rest of its portions. So that light staining central portion of the A band is called the H zone. Enclosed in the red rectangle is the H zone of the A band. That's the portion of the A band that appears lighter than the other portions. It's very obvious in the picture that this H zone would appear lighter because you only have in there the thick filaments, while you have both the actin and myosin, the thin and the thick filaments respectively, in the other portions of the A bands. So if you will be asked in the exam what filament or filaments is or are present in the H zone, you should only answer thick or myosin filaments. But if you will be asked what filament or filaments is or are present in the A band, you should answer both actin and myosin. The structure that divides the A band in the center is called the M line. And this M line is composed of the protein myosin. The green-colored spring-like structure that links the myosin to the Z line is called the titin. And its main purpose is to stabilize the thick filament, and it prevents the overstretching of the sarcomere. During muscle contraction, the myosin filaments will pull the actin filaments towards the M line, thereby shortening the sarcomere. But as soon as the muscle will relax, the sarcomere will go back to its original size. But for the sarcomere not to overstretch, the myosin is attached to the Z line by the protein we call as the titin. If we will cut through this portion of the sarcomere, we're cutting through its A band. You have there the presence of the thick and thin filaments. And this is how the filaments are arranged. The small red circles are representing the thin filaments or the actin, while the blue circles are representing the myosin or thick filaments. What do you notice? One myosin is surrounded by 6 actin filaments. If we will cut through this portion of the sarcomere, we're cutting through its H zone, and expect that what you only have in there are the myosin filaments. If we will cut through this portion of the sarcomere, we're cutting through its M line. If we will cut through this portion of the sarcomere, we're cutting through its I band. So expect that what you will only see in there are the red-colored thin filaments or actin. The small green circles are representing the titin proteins. Remember, they are there to attach the myosin to the Z disc. At this point, we will be discussing the thick and the thin filaments. Shown in the picture are actin, the thin filament, and myosin, the thick filament. Take a look at the purple-colored myosin. It is very obvious in the picture that these myosin filaments are bundled together, and they have tails and globular heads. You have already learned that the myosin or the thick filament is found in the A band, and it is only the filament that we can find in the H zone of the A band. Myosin is composed of myosin filaments that are bundled together, and they have tails and heads. Take a look at the heads of the myosin. They are oriented towards the actin filaments. Please remember that the myosin filament is surrounded by 6 actin molecules. That's why you can see that the heads of these myosin filaments are oriented all throughout the circumference of the myosin filament because they are surrounded by the 6 actin filaments. This red arrow is showing or pointing the tails of the myosin filaments. I intentionally inserted this slide for you to appreciate that the heads of the myosin filaments are oriented towards the actin or the thin filaments because they have corresponding binding sites in the actin filaments. And the binding of the heads of the myosin filaments to the actin filaments will now facilitate the shortening of the sarcomere during muscular contraction. In the A-band, you have two myosin filaments on both sides and their tails are connected to the center, or we call it as the M line. And the M line is composed of myosin. And these myosin filaments are also connected to the Z disc by the green-colored spring-like structure we call as the titin proteins. Let's now move to the thin or the actin filament. This type of filament is present in both I-band and A-bands except for the H zone of the A-band because in the H zone you only have there the myosin filament. Now, one end of the thin filament or actin filament is attached to the Z disc, while its other end is found on the A-band, particularly at the edge or border of the H zone. So meaning, the entire actin filament is from one Z disc up to the border of the H zone. Again, one end of the thin or actin filament is attached to the Z line and the other end is present in the A band up to the border of the H zone. What we will do at this point is to highlight one thin filament and look at its individual parts. We will now discuss the individual components of the actin or the thin filament. Let's start with this structure pointed by the red arrow. That one spherical structure is called the G-actin. The G letter there stands for the word globular because the structure appears to be globular in shape. And then, can you appreciate the presence of a depression within the G-actin? That is now the binding site for the head of myosin. Now, if you group G-actins, they will form the whole F-actin. So the entire picture is actually one F-actin. F stands for filamentous actin. One globular structure, G-actin. Globular structures grouped together, filamentous actin or F-actin. And then, there is that thread-like structure which covers the binding site of myosin in G-actin. That thread-like structure is called the tropomyosin. So it's actually there to inhibit the binding of myosin to the actin, especially during muscular relaxation. To help the tropomyosin regulate the binding of myosin to the binding site in G-actin or the F-actin, the tropomyosin will be helped by these three circular structures and we collectively call them as the troponin complex. And they have special names. They are troponin T, troponin C, and troponin I. The individual circular or globular structures making up the actin are called G-actin. If these G-actins are grouped together, they will now form the filamentous or F-actin. During muscle contraction, it's very important that the myosin head will be able to bind to the binding sites present in the actin. But if the muscle is relaxed, those binding sites should not be exposed and they should be covered by the tropomyosin with the help of the troponin complex, which is composed of what again? Troponin C, troponin T, and troponin I. The tropomyosin is a protein that wraps around the binding site in actin and prevents the myosin from binding to it. In a relaxed muscle, the tropomyosin blocks the attachment site for the myosin so that the myosin cannot do the cross-bridging with actin, thereby preventing contraction. Troponins are proteins attached to the tropomyosin, and they somehow help in inhibiting muscular contraction. And this troponin complex is made up of troponin T, troponin I, troponin C. For you to easily memorize the parts of myosin and actin filaments, I want you to think of myosin as the guy or the suitor. Myosin, mayaman. And then, the girl is the actin. And the girl has the holes for the binding site of myosin. Unfortunately, actin has a very strict mother. And the name of the mother is tropomyosin. And tropomyosin functions to cover the holes of actin. And to add to that, actin has three big brothers. And we call them as troponins C, T, and I. And along with the mother, tropomyosin, they will also inhibit the binding of myosin to the holes of actin. The little girl with the holes is actin, and she has a strict mother, and the name of the mother is tropomyosin, and mother is covering the holes of little girl. Little girl has three big brothers, the first of which is troponin I. So I want you to take a look at the location of the troponin I. It's located between the troponin complex and the actin. So what will bind the troponin complex to the actin is troponin I. And then the other big brother is troponin C. Okay, I will explain the role of troponin C later on. The other big brother is named troponin T. And I want you to take a look at that troponin T. It interacts with the tropomyosin. So therefore, what will bind the troponin complex to the tropomyosin is troponin T. So that's the reason why it's called troponin T. The T there somehow refers to its binding to tropomyosin. Of the three components of the troponin complex, it is the troponin I that is bound to the G-actin. So the main role of troponin I is to bind the troponin complex to the G-actin. And since the troponin complex is bound also to tropomyosin via the troponin T, so troponin I functions to hold together the actin, troponin, and tropomyosin complex. The other member of the troponin complex is troponin T, and it is named troponin T because it binds the whole troponin complex to the tropomyosin. The last component of the troponin complex is the troponin C. It's named such because it has binding sites for calcium. And you know what will happen after calcium will bind to the troponin C? The troponin C will undergo conformational change. You can just simply imagine it as troponin C changing its shape. And you know this change in the conformation or shape of troponin C will facilitate later on muscular contraction. How? I will explain that in the next succeeding slides. So I want you to take a look at this picture. This is tropomyosin, and look at how it interacts with the G-actins of the thin filament. This is your troponin T. Okay, take a look at its attachment to the tropomyosin. And this is your troponin C. Can you appreciate that it has this binding site for calcium? And this is your troponin I. It's particularly attached to the troponin complex, then directly to the G-actin of the thin filament. This is the troponin C that is not yet bound to calcium. And once calcium will bind to troponin, the troponin will undergo conformational change or change in shape. So I want you to imagine if the troponin C will change in shape, it will become twisted, it will become smaller, whatever is the change. The thing is, it will lead to the pulling of troponin T. If troponin T is pulled by troponin C, troponin T in return will pull tropomyosin. So the change in the shape of troponin C will cause the displacement of troponin T and tropomyosin. And this displacement will now cause the exposure of the binding sites of myosin in the actin. So the myosin now can readily bind to the binding sites in the actin. So for you to imagine that, I want you to think of troponin C as the big brother who can be bribed with money. You know very well that troponins C, T, and I, and tropomyosin are supposed to inhibit the binding of myosin to actin by covering the binding sites in actin. But the thing is, in the presence of cash, Troponin C will change his mind. And what will happen? If Troponin C will change his mind, he will pull big brother Troponin T. And then, later on, Troponin T will pull mother tropomyosin, exposing now the holes in actin. That's why myosin is now free to bind to the holes or binding sites in actin. In photo A, all the binding sites for myosin are blocked by the troponin-tropomyosin complexes. In letter B, you have there the presence of calcium ions, and it's very obvious in the picture that the calcium ions bind to one of the troponins, and that's troponin C. And the binding of calcium ions to troponin C will later cause the troponin and tropomyosin complexes to be displaced, exposing now the binding sites in G actin for myosin. With the binding sites exposed, the heads of the myosin now can readily bind to them, causing now cross-bridging, eventually leading to the shortening of the sarcomere and the muscular contraction. The calcium ions needed to cause the conformational change in the troponin C to facilitate muscular contraction are stored in sarcoplasmic reticulum. This is the organelle in muscles that is equivalent to the smooth endoplasmic reticulum. In a relaxed muscle, the calcium ions are all stored in sarcoplasmic reticulum. But with the presence of stimulation from the nerves, the sarcoplasmic reticulum will release these calcium ions, and the calcium ions will bind to troponin C. You have here in this picture the plasma membrane of muscle fibers. That's the sarcolemma. Then, I want you to appreciate that this sarcolemma will have finger-like extensions towards the muscle fibers, and these finger-like projections are called the T-tubules. Then, on both sides of the T-tubules, you have there the terminal cisternae of the sarcoplasmic reticulum. Since you have three structures in one complex, you will now call them as a triad. So the triad is composed of the T-tubule, which is the extension of the sarcolemma into the muscle fibers, and the terminal cisternae of the sarcoplasmic reticulum. Take a look at the nerve in the picture. The nerve has released acetylcholine neurotransmitter into the synaptic cleft. The acetylcholine molecules will bind to the corresponding receptors on the sarcolemma of the muscle. Now, the binding of acetylcholine into the receptors of the sarcolemma will produce muscular impulses. And since the T-tubules, the ones labeled with number 2 in the picture, are extensions of the sarcolemma, these muscular impulses will also travel towards the T-tubules. And do you know what will happen? The muscular impulse will now activate the terminal cisternae of the sarcoplasmic reticula on both sides of the T-tubules. And the activation of the terminal cisternae of the sarcoplasmic reticula will lead to the release of the calcium ions. So this photo is just showing us how the nerve will be able to make the muscle produce a contraction. So again, the T-tubules, or also known as the transverse tubules, are extensions of the cell membrane that will invaginate or penetrate into the skeletal or cardiac muscle. Okay? So I hope you have already memorized the components that make up the triad. So take a look at this picture. This is the sarcolemma. This is the extension of the sarcolemma, which will eventually become the T-tubule. And I want you to appreciate that besides each T-tubule, you have the terminal cisternae of the sarcoplasmic reticulum. As the muscles contract, there is shortening of the sarcomeres. And this is primarily because of the pulling of actin filaments by the myosin filaments towards the M line. And this entire mechanism is referred to as the sliding filament. Let's discuss the processes happening in sliding filament mechanism. Let's start with number 1. You have there the binding of calcium into the binding sites in troponin C, causing now the change in the shape of troponin C, later leading to the displacement of the troponin and tropomyosin complex, exposing now the binding sites of myosin in the actin filaments. What will happen next is the myosin head will now bind to the binding sites in the actin. And I want you to take a look at the myosin head. It is bound to ADP and phosphate. The next thing that will happen is the ADP and the phosphate will be released from the myosin head, and the loss of ADP and phosphate will cause the head to bend. And the bending of the head will now cause what we call as the power stroke. So the power stroke is the one responsible for the pulling of the actin filaments towards the center by the myosin filaments. And you know what will happen after? After the power stroke, one ATP molecule will bind to the myosin head, causing now the myosin head to detach from the actin filament. The hydrolysis of the ATP that is bound to the head of myosin will result to the production of ADP and phosphate, and the hydrolysis of ATP will now cause the head to return to its original position. And you know what will happen after? The head will bind to the binding sites of actin and create another round of power stroke. And these processes will keep on repeating until such time that the sarcomere will become short and it will now be seen visually as a muscular contraction. That's why there is really shortening of sarcomeres during muscular contraction. One of the post-mortem changes that we can observe in dead people or cadavers is the stiffening of the body muscles, and this is referred to as the rigor mortis. And the reason behind it is that if the person is dead, he stops producing ATP. And what will cause the myosin head to detach from the actin binding site after generating the power stroke is ATP. So if ATP is not present anymore, the myosin will no longer detach from the binding site in actin, causing now the stiffening of the body muscles. Let us now discuss the changes in the sarcomere during muscular contraction. This is the I-band. This is just half of the I-band because you have there the border which is ZDis, and ZDis is found in the center of one I-band. But what I want you to take note is that during muscular contraction, during the shortening of the sarcomere, this I-band will disappear. And then, I also want you to take note that the H-zone during muscular contraction will become narrow. But the A-band will remain the same. So what are now the changes in sliding filament mechanism? The H-band will become narrow, the I-band will disappear, while the A-band will still remain the same. During muscular contraction, the I-band will disappear, the H-zone will disappear, the A-band will remain the same, and the length of the sarcomere will become shorter. So I'll just give you 10 seconds to read all the things that are posted in this PPT slide. Do you know that there are different types of skeletal muscles based on the number of mitochondria present in the muscle fibers and based on the concentration of their myoglobin? You are very much aware that myoglobin is the pigment responsible for the red color of muscle fibers, and mitochondria are the powerhouses of the cell. let's try to review the importance of mitochondria and myoglobin. So take a look at the picture of the mitochondrion. You have their pyruvate molecule generated from glycolysis in cytoplasm. This pyruvate will enter the mitochondrion and is converted to acetyl coenzyme A. This acetyl coenzyme A will participate in the Krebs cycle. So I want you to focus on the right photo. One of the products of the Krebs cycle or the citric acid cycle is NADH. And this NADH will proceed to the next physiologic process, and we call it as the electron transport chain. Do you know what will happen in electron transport chain? The electron in NADH is transmitted to the proteins that participate in the electron transport chain. And these electrons will be transferred from one protein to the other. Once the electron will now reach the last protein of the electron transport chain, something must accept the electron from that last protein. And that's something I am pointing to is the oxygen. So the last acceptor of electron to complete the process of electron transport chain is oxygen. And what will provide this oxygen is the myoglobin. So the point is, if a skeletal muscle is rich in mitochondria and myoglobin, that particular muscle can have abundant production of ATP because they have mitochondria to facilitate the Krebs cycle and electron transport chain, and they can complete the electron transport chain because they are provided with abundant myoglobin pigment. The first type of skeletal muscle is the type 1 or the slow red oxidative fiber, and I will explain to you later on why they are described that way. Red because they are rich in myoglobin. And these skeletal muscles are also rich in mitochondria. So just try to imagine the number of ATP molecules that this type of skeletal muscles can produce. So they will never ever be subjected to fatigue. And they can sustain the contraction because they can always produce ATP. But the thing is, since the entire process of ATP production in the mitochondria would take time, just try to remember, glucose undergoes glycolysis to produce pyruvate, pyruvate entering mitochondrion to become acetyl coenzyme A, acetyl coenzyme A entering the Krebs cycle. The Krebs cycle will produce NADH and FADH2. The electrons of these two products will transfer to the electron transport chain, and the last acceptor would be the oxygen. So just try to imagine how lengthy the process is. So that's why, since the production of ATP is a slow one, expect that the contraction that can be generated by this group of skeletal muscles will also be slow, but sustained. Why? Because they can always generate ATP. Good examples of these skeletal muscles are the skeletal muscles responsible for maintaining our posture. Example, the back muscles. Okay, they can sustain the contraction to keep us in the sitting or standing position without us feeling the fatigue. But these muscles cannot generate contractions as fast as the muscles of the fingers or the digits, but they can sustain the contraction. So the second type of skeletal muscle is type 2B, and they are named fast white glycolytic fiber. White because they have less myoglobin, and they also have less abundant mitochondria. But what makes them white is the presence of abundant glycogen. So since they have glycogen stores, they can easily break them down to produce energy. These muscles can generate fast contractions. But since they don't have enough mitochondria, they don't have enough myoglobin, once their glycogen stores will run out, this type of muscles will be subjected to fatigue. Yes, they might generate contractions faster than type 1, but they cannot sustain them. Good examples of these muscles are the muscles that move the eyes and the muscles of the digits. So examples of type 2B are the muscles that move the eye and the muscles of the finger. They can contract faster than the postural muscles, but they are easily subjected to fatigue. Type 2A skeletal muscles are intermediate between type 1 and type 2B skeletal muscles, and they are described to be fast, intermediate, oxidative glycolytic fibers. Oxidative because they have myoglobin, pigments, and mitochondria, but not as much as type 1. Glycolytic because they have glycogen stores, but not as abundant as type 2B. That's why these muscles are said to be using both oxidative metabolism, similar to type 1, because you have their myoglobin supplying oxygen to complete the entire process of electron transport chain, and anaerobic glycolysis because they can also break down the glycogen stores without relying much on the electron transport chain, similar to type 1 skeletal muscles. And this type 2A are adapted for rapid contractions and short bursts of activity. That's why these are the muscles that most athletes would want to develop. The fiber on the left side of the screen is a type 1 skeletal muscle. I'm pretty sure because it has the highest myoglobin content. While the muscle on the right side of the screen is type 2B because it is composed of white fibers, and these white fibers are composed of less of myoglobin and more of glycogen stores. And the muscle in the middle is the intermediate of type 1 and type 2B skeletal muscle. So that is the type 2A. So this is the summary of the differences among type 1, type 2A, and type 2B skeletal muscles. In terms of mitochondrial density and myoglobin concentration, type 1 has the highest while type 2B has the lowest. In terms of the speed of contraction, type 2B generates very fast muscular contraction while type 1 is slowest in terms of the speed of contraction time. Then, in terms of resistance to fatigue, this will be dependent on the mitochondrial density and myoglobin pigment. So expect that type 1 would be the type of skeletal muscle that would have the highest resistance to fatigue. In non-athletic individuals just like me, there is that 50-50 ratio of type 1 and type 2 skeletal muscles. You will only see the significant difference in the numbers of these type 1 and type 2 skeletal muscle fibers in athletic individuals. Power athletes, just like the splinters, would usually have more of type 2 skeletal muscles than type 1 because they need to run faster compared to their competitors. While endurance athletes, those that are participating in marathons or those that are considered to be distance runners, they don't have to run very fast because the main goal is to reach the finish line. So for these people to sustain the contraction from the start down to the finish line, they need to have more of type 1 skeletal muscles than type 2. The next topic that we will discuss is all about the muscle spindle and the tendon organs. Let us first start with the muscle spindle. Muscle spindles are structures inserted among the fascicles of the skeletal muscles. And it's easy to identify the muscle spindles because they are covered with capsule. So take a look at this red arrow. It's pointing to one muscle spindle. It's very obvious in the picture that that muscle spindle is surrounded by fascicles of skeletal muscles. Now take a look at the white covering, that is the capsule. Now this muscle spindle is composed of specialized type of skeletal muscles. Now, the skeletal muscles that we can find within the muscle spindle should be referred to as intrafusal fibers. While the ordinary skeletal muscles that we can find outside the muscle spindle or within the fascicles of the skeletal muscles will be referred to as extrafusal fibers. Again, the skeletal muscles within the muscle spindle are called intrafusal fibers, while the muscles within the fascicles, the ordinary skeletal muscles, we refer to them as the extrafusal fibers. Now, what is now or are the purposes of muscle spindles? Number one, it detects distension or stretch of the muscle caused by body movement. I will explain this one by giving you an example later on. Number two, these muscle spindles are also proprioceptors, meaning they can detect positions of the muscle. So I want you to close your eyes right now and move your hand in front of you. You can still determine the position of your hand. It's because the muscle spindles are telling you the position. And that's what we mean by the muscle spindles acting as proprioceptors. Take a look at this person. This person is bending his body towards the right side. And what will happen if he will continuously do it, he might lose balance. This bending of the body towards the right side will cause the muscles on the left side of the body to be stretched. And do you know the function of the muscle spindles? The stretching of these muscles will cause the muscle spindles to be stretched. And the muscle spindles will send a message to the brain that your body is bending towards the right side and you might lose balance. So that's why the muscle spindle functions to detect distension or stretching of muscles. Aside from that, muscle spindles also act as proprioceptors. They can determine the position of the muscle. So even if you are blindfolded, you can still determine the position of your legs and hand. So let's try to identify muscle spindles under the microscope. These four arrows are pointing to fascicles. Fascicles are grouped of muscle fibers. And then, take a look at the muscle fibers. They are multinucleated and their nuclei are found at the periphery. So these are skeletal muscles. Found between the fascicles is a capsulated structure. So this is the muscle spindle. And the muscle fibers in its central portion are called the intrafusal fibers, while the muscle fibers found within the four fascicles are called the extrafusal fibers. Blue arrow is pointing to one fascicle, so you have there intrafusal fibers. Red arrow is pointing to, yes, a muscle spindle because it is covered with a capsule. So the muscle fibers inside it are called intrafusal fibers. Blue arrow is pointing to one fascicle. So the muscle fiber pointed by the blue arrow is an extrafusal fiber, while the red arrow is pointing to the capsulated muscle spindle. So the skeletal muscle fibers found within it are called intrafusal fibers. The next structure that we will discuss is the Golgi Tendon. From the name of the structure, you can already predict where we can find it. The tendon is the dense regular connective tissue that attaches the muscle to the bone. Now, what is this Golgi Tendon? This Golgi Tendon is composed of group of axons innervating the collagen bundles in the tendon. You know very well that the tendon is composed of dense regular connective tissue. When you say dense regular connective tissue, it is composed of abundant collagen fibers. So if these collagen fibers will be innervated by a group of axons, they will now form what we call the Golgi tendon. What is now the function of the Golgi tendon? It is positioned in the tendon to detect tension if the tendon will be pulled by the muscle, especially if we are bearing heavy weights. So I want you to imagine you are trying to carry something that is heavy. So you are generating powerful muscular contraction and the muscle will be creating tension on the tendon. To avoid damage to the muscle because of excessive contraction, the Golgi tendon functions to detect the limit of the tension, and this Golgi tendon will inhibit the motor nerve controlling that muscle so that the muscle will no longer contract and it will now lead to the trauma or injury of that muscle. Take a look at the Golgi tendon in the photo. So if this Golgi tendon will detect excessive distension of the muscle, it will activate a reflex. And I want you to follow the black arrows. So this Golgi tendon will activate an inhibitory interneuron in the spinal cord. And from the name itself, this interneuron will inhibit the motor neuron that is causing that muscle to contract. So what will happen? The motor neuron will be inhibited and the muscle will stop contracting further. There is a chance that if a weightlifter will accidentally drop the weight that he's supposed to carry, that the Golgi tendon was activated, and the Golgi tendon activated the inhibitory neuron, and the inhibitory neuron inhibited the motor neuron that is making the muscle undergo contraction. So let's discuss the physiology of smooth muscle. So appreciate first the shape of the smooth muscle. It is spindle in shape or fusiform in shape. And then it has a lot of gap junctions on the surface or the cell membrane. And you know the function of the gap junction, right? It allows exchange of chemicals between two adjacent smooth muscles. And then I want you to appreciate the sarcolemma of the smooth muscle. Can you appreciate small invaginations? These small invaginations are called caveolae. Okay? I want you to take note that for this smooth muscle to contract, it should allow calcium ions to enter its sarcoplasm or cytoplasm. And for more calcium to enter the cell, okay, there must be an increased surface area. And the main purpose of the caveolae is to actually increase the surface area of the sarcolemma of this smooth muscle so that there would be higher number of calcium ions that would enter the muscle cell. Do you know that smooth muscles also have myosin and actin filaments? So how come they are not striated similar to skeletal and cardiac muscles? Remember, in the sarcomeres of skeletal and cardiac muscles, the myosin and actin filaments are arranged in a particular pattern. That's why they can give these muscles the appearance to have alternating light and dark bands. Thus, the muscles appear to be striated. In the case of smooth muscles, the myosin and actin filaments are not arranged in that particular pattern, so they cannot make the muscle appear to be striated. Do you know that there are structures that are present in smooth muscles that are not seen in skeletal and cardiac muscles, and we will discuss them one by one. But first, I want you to take a look at the actin and myosin filaments. So highlighted on the 12 or 1 o'clock position of the photo, the picture of the actin and myosin filaments. So during muscular contraction, the myosin will pull the actin filaments towards the center. One unique structure in smooth muscle is the dense body. This is the point of attachment of the actin filaments. So expect that if the actin filaments are pulled towards the center by myosin, the actin filaments will be pulling the dense bodies. Do you know that the smooth muscles also have a third type of filament? So in skeletal and cardiac, we only have two, the actin and myosin. In the case of smooth muscles, they have a third type of filament, and that is the intermediate filament. So I want you to take a look at the brown-colored intermediate filament. It is attached to the dense body, and at the same time, it is also attached to the sarcolemma or cell membrane of the smooth muscle. So I want you to imagine, during muscular contraction, the myosin will pull the actin filaments towards the center. The actin filaments will pull the dense bodies. The dense bodies in return will pull the intermediate filaments. And since these intermediate filaments are attached to the sarcolemma, they will also pull the cell membrane of the smooth muscle, causing now the shortening of the individual smooth muscle fiber and generating now contraction. So if the myosin will bind to actin, both of them will be pulling the dense bodies. The dense bodies will be pulling the intermediate filaments, causing now the contracted state of the smooth muscle. So this is just to show you how smooth muscles will contract secondary to the interaction of myosin to the actin, causing now the pulling of the dense bodies and the intermediate filaments. Again, smooth muscles contain myosin and actin filaments, and these filaments are attached to dense bodies. The myosin and actin filaments have less regular arrangement. That's why they cannot make the smooth muscle appear to be striated. And there are no troponin and tropomyosin molecules present in the smooth muscles. Instead, these smooth muscles have dense bodies and intermediate filaments. The smooth muscles are under the control of the autonomic nervous system. So the nerves of the autonomic nervous system will activate these smooth muscles to contract by releasing neurotransmitters. And these neurotransmitters will act on the smooth muscles by allowing the entry of calcium through the receptors in the sarcolemma or in the cavulae. The entry of calcium ions into the sarcoplasm or cytoplasm of the smooth muscles will activate the sarcoplasmic reticulum to release the stored or sequestered calcium. And the main purpose of which is to increase the calcium ions in the sarcoplasm of the smooth muscle. Now, these calcium ions will bind to calmodulin. So they will form a complex, and the complex is called calcium-calmodulin complex. This complex can activate a particular enzyme, and that enzyme is myosin light chain kinase. Whenever we think of the kinase enzyme, it has something to do with catalyzing reactions that involve the transfer of phosphate group. In the case of myosin light chain kinase, it transferred the phosphate group of ATP, one phosphate group, from ATP to the myosin filaments. And the myosin filaments now will become activated. So these myosin filaments will only become active if they are phosphorylated. Now, once phosphorylated, these myosin filaments will bind to the actin filaments, causing now the contraction. Remember, there are no troponin and tropomyosin proteins in the actin filaments of the smooth muscles. So, the requirement is for the myosin filament to be phosphorylated. So that ends my discussion on the part 2 of muscle tissue histology. Thank you for listening, future doctors.